Also, the final part of our regulating system is sRNA. Unlike the two that had already introduced, light and temperature regulation, sRNA is not an stimulation dictator, but an inhibitor that produced by the bacteria itself. When an uncoded sRNA is transcripted, it would recognize a special sequence. Here we call it sRNA binding site, which includes the Shantagano sequence. And the sRNA would target to this special sequence by base pairing. Once the sRNA attached to the mRNA, it would block the ribosome binding site. Therefore, the ribosome could not attach to the mRNA and no protein would be translated. So we first chose two artificial sRNA that are originally from the E. coli and mutated a few points in the sRNA target region shown gray in the structure on this slide. This is because we want them to work in E. coli but won't affect the bacterial growth or other genes that we put in. Next, we synthesize the sRNA binding site according to the reverse and complementary sequence of the sRNA target region. Let's take a quick view of how the sRNA system works when becoming a biobrick. So, if we assemble a circuit like this, we put the sRNA binding site in front of the gene that we are interested in. Take red forest protein for example. Because the sRNA binding site includes the sequence of Shantagano region, the RFP could be translated normally. But when we put the sRNA into our E. coli, it would bind to the sRNA binding site and keeping the RFP and shut down the RFP gene translated. So in this mechanism, our sRNA can stop our gene expression at RNA level. What is the advantage of using sRNA regulation? As you can see in this circuit, if our cultural condition is under 37 degrees, only RFP could be translated. With the temperature rising to 37 degrees, the 37 degrees RBS starts to work, making its downstream gene GFP to be translated. But the common RBS does not lose its function when the temperature rises, which means, in this case, about 37 degrees, we both have red fluorescence and green fluorescence expression. Now, let's combine the sRNA system and the temperature regulating. Here, we replace the common RBS with the sRNA binding site and add a LUX operon to induce the production of our sRNA. When the temperature is below 37 degrees, the RFP could translate normally. With the temperature rises, the GFP and LUXR genes start to express. With the presence of AHL, LUXR protein can induce the transcription of sRNA, which is at the downstream of PLUX, and it will inhibit the RFP translation. So, in this case, we can have only one product at one temperature. In addition to making us more easily to switch between several products and different conditions, the sRNA can also give us more choice to regulate our gene expression. Unlike the promoter regulation we often use, for example, tacopron, necopron, and luxopron, which can only use one time in one project. We can put several different sRNA parts in our project, and by combining different promoters to different sRNA, we can create more specific or more regulation system to manipulate our bacterial robot. So now you understand our concept of regulating system. Let's take a look at our overview project.